Hi everyone, thank you for joining this presentation. And I would also like to thank the organizing committee for the warm invitation to represent Bluu Seafood at this Space Summit. And what I want to present to you today is how fresh seafood in space can be made possible by Bluu. My name is Young, and I'm the Director of Research and Process Innovation at Bluu. In essence, I currently lead the team that does development for how do we make or how do we mass produce the fish cells for our various seafood products. All right, so before we start the topic of cultivated seafood, I want to also talk about alternative proteins from other methods. The first one would be plant-based alternative protein, and these are directly produced from plants, and they provide nutrition such as protein, fat, vitamins, and minerals. A second method to produce alternative proteins is through fermentation of microorganisms such as yeast and fungi. And these two products actually you can already find in your local grocery store. Then the third one is cultivated proteins, and these are produced from cells. The cells are actually isolated from the animal itself, either be a pig, a cow, or a fish. And then these cells are actually, let's say, produced in a big fermenter, and from that you would get the proteins you need, and they're from eating the cells themselves. One of the reasons why Bluu was found is to protect our ecosystem, the marine ecosystem specifically. And that is because the current fishing industry is destroying oceans uh, and having heavy environmental impacts. There's even health issues with the fish that you consume due to microplastics and the cruelty that goes with the big kind of net catching of the fish, where the bycatch fish are also being slaughtered, but they can't be consumed. And for the environmental factor, the CO2 emissions from these vessels, the fishing vessels, is also just destroying the oceans. And the fishing that is being done is actually overfishing, and we are maxing out the capacity that the ocean can actually provide because the demand for seafood is just going up and up. As you can see in the graph on the right here, that the current demand for seafood is equivalent to about 500 billion US dollars in sales. And in about 10 or so years, you can see that it will double in the amount of demand that's needed. And if you compare to the other animal proteins on the graph here on the left, you can see that seafood takes up 35% of consumption compared to beef alone and pork alone. Even chicken. Chicken is still only at 26% compared to seafood. So the cultivated seafood industry is trying to match or at least provide the correct supply that this growing population is demanding for seafood. And there are many players around the world that does cultivated seafood. However, Bluu Seafood is the first one in Europe and the biggest one in Europe. And we plan to pave the way for EU to be the first one as well to have a product on the market. Since the day that Bluu was founded, we focus on creating a cell technology expertise and a food technology expertise to create the first product as quickly as possible. And the fish balls is one of our first products that have already been press released. And the fish ball is an unstructured product. So what unstructured means is that the cell biomass that we produce becomes an ingredient in a hybrid recipe for the fish ball. The hybrid recipe includes both the fish biomass and some plant-based ingredients. And the plant-based ingredients helps with providing a little bit more texture and mouthfeel, while the fish flavor itself comes mostly from the fish cell biomass. Our next product would be fish fingers, and this one would have a structured flaky texture, similar to what you would find, on a fish stick in the freezer aisle as well. And then our third product would be small pieces of structured muscle uh, pieces, and this could be used to form, for example, a tartare. And then the long-term goal and the holy grail milestone would be having a whole salmon fillet. And this is fully structured with muscle cells and fat cells combined together so that the texture, mouthfeel, and flavor all come from the fish cell biomass, the differentiated fish cell biomass. So how do we even produce this fish cell biomass? And here the slide shows the process that we are using at the moment. The first step is to isolate the cells and create an immortal cell line. So we take samples from the fish and we cultivate them in a 2D surface or surface itself to expand them to be more than 10 or 20 cells. We need hundreds or millions of cells, and the cells actually will go through a spontaneous immortalization. That means they will spontaneously go and become immortal, meaning that they can divide indefinitely for a lot of different passages or time to come. The second step is to then get these cells here that are adherent to a 2D surface to grow into a suspension. So they should be floating around in the same nutritious broth or medium that they really like. And why do we do this? We do this to have the possibility of scaling up. If the cells are always adherent and in 2D, the scaling up process will be very challenging and will take a lot of resources compared to cells that are growing into suspension instead. And the suspension cells look, for example, like this, small aggregates or spheroids, because they still have the tendency to stick to each other, or that they like neighbors around them so that they will survive and be happy. So with the cells in suspension like this, we scale up to bigger bioreactors, such as a stir tank bioreactor. And with the stir tank bioreactor, we can go from, for example, a 500 milliliter bioreactor to 5,000, 10,000, or 20,000 liter bioreactors. That is still based on the stirring capacity. And in order to achieve all this, we need a lot of expertise. And to go for the long-term goal of a fillet, it looks simple from our perspective, but if you dive deep and get into the details or the microscopic details of a fillet, it's very complex. So then we need various expertise to accomplish the long-term goal of a fish fillet. And that is through expertise in media development. So we would need biochemistry, molecular biology to figure out what is the best nutritional broth that these cells would like. Not only is it different for salmon, for example, but also for every different types of fish, you would need a different nutritional recipe. Then the cell line development team, which is made of cell biologists, and what they do is they figure out how to best get or isolate the cells that would be great for this kind of process. And once they have that cell, they create a lot of different cell lines, and then they bank the cell lines so that everything is always in reserve and we don't have to go back to the fish again and again. So once we have the cells and the nutritional broth, we think about the bioprocess development. And this is where, we take like 1 million cells and we do 100 times fold in the biomass production, or even 1,000-fold. And so we not only produce the proliferating cell biomass, but we will also look into how to create a process for differentiating these stem cells into muscle and fat, because the muscle cells give the highest nutrition, and also the fat cells, because of the omega-3s. And with the fat cells as well, you get a lot of flavor as you cook, and after you cook, that's what you would want to taste. It also gives you this moisture content that's very similar to the natural fillet. And all this puts together, there's a product development team where we have food scientists and food technologists. They formulate the recipes, they formulate the storage conditions, the packaging, the method of cooking, so that the consumer can easily purchase something from the market and prepare it for their families at home. So to dive deeper into the cell line development, as you can see in this image here, a cross section of a fish, and you can see that there are different sections of the fish, and within these different sections you have different types of muscle and different types of fat. And so the goal or the expertise of the cell line team is to be able to isolate the cells specific to fat or muscle, only the progenitor cells, and they have to figure out which method is best for which type of fish, because there are fish that have more fatty tissue and then there's fish that have more lean tissue, so these processes will be different. And the location of the tissue to get the best progenitor type of cells, and the method for culturing them so that we get the best immortal cells to then proliferate or expand them to be cell lines. Then we check for cell type specificity. So we need to know that we're isolating only progenitor fat cells, only progenitor muscle cells, and then checking methods to even optimize or improve immortalization, rather than just counting on the spontaneous part of the cells. And as you can see here, you can do various different characterization to visualize if you have isolated the correct cells. So here you can see that the trout biomarker, or a specific protein inside the cell that says, yes, I am a progenitor muscle cell or a muscle stem cell. Same with here, you can see that more clearly, or with a lot more cells, that the red is indicating that there are stem cells that can become muscle. And that's part of the cell type characterization. And we need to really identify that these cells can either be fat or muscle. And depending on the species, this is very important as well. And since we are growing the cells to many, many passages or infinitely, we need to check that they have stability in their genetic information and that the cells are homogeneous in their population. So with all the cells being characterized, then they are ready for banking. And banking is just about reserving or freezing down cells so that there are a lot of frozen vials in which we can just use them, those cells, instead of going back to the fish to collect more cells. So once we have the cell lines, we have to figure out what is the nutritional need that these cells need. And first we have to think about, are there formulas that would be fish specific? What kind of salts do we need to add in there? Proteins, amino acids, other growth factors or proteins, some kind of acid base to balance the pH, some sugars, and possibly some other growth factors and proteins that the fish cells also need. And once we figure that out, we also, in the meantime, we also need to know that we can source these components in a food grade manner, or have a certificate in which they're food grade or safe for consumption. The biggest challenge actually for every company is to replace or remove this fetal bovine serum in the culture nutrition medium. And once we are able to do food grade components and replace the fetal bovine serum, the reduction in costs will be easily done. And by reducing the cost of the medium, we also then can be competitive to the current prices of subsidized fish. So once we have the cells and the media broth, we have to figure out how to get these cells into suspension so that the cells are growing in just suspended in a liquid solution. But as you realize, fish cells naturally don't grow in this kind of floating situation. So we do have to control a lot of parameters and also get them to adapt to this condition. And they actually can adapt, and that's part of the phenomenon or the uh, capacity of cell biology. So once they have adapted and we found the right conditions in which they are proliferating, we can then increase the yield by even optimizing that further. And we also have to figure out how to do this in a food safe process. So all the solutions that we use in the upstream and downstream, and then the storage of the product at the end. And the product here is the cell biomass. How do we store it to keep its nutrition, to keep its flavor, and to keep its like food technological properties? And once we have this as well, we can start increasing the scale of how much we can produce, going from the 1 liter to 50,000 liters. And that can be done through a lot of automation and thinking about the process with ease of handling. So after bioprocessing, we then have a fish cell biomass ingredient. So then the product development team, what they do is develop recipes that incorporate or test the fish flavor, the mouthfeel, the texture, and the aftertaste. And their main goal is to think about food and how food is experienced by the consumer. So ordinarily when you have food, you will look at the food and see its color. Then you would smell how it's cooked or when it's cooking, even at the raw state, how is the odor? And when it's cooked, after it's cooked, you want to have a very inviting, delicious smell to the seafood. And lastly, you want to be able to taste what it is that you should be eating. So if you're eating the fish balls, it should have fishy flavor, but if you're eating the fish fillet, it should have even more fish flavor. And then the texture, for example, between the fish ball and the fish fillet definitely should be different. Not only the texture just in your mouth as you chew, but also the texture as you cut it open with the fork or your knife, and kind of visual texture as well. And what they do is, within the recipe development, is to do a lot of different taste testing. And they create one recipe, they do a tasting. Is it good? Is it bad? Where is it good? Where is it bad? And then they go back and reformulate the recipe. They can also do this with instrumental sensory testing, so a texture analysis, or even an instrument that can mimic the smell of a nose to see if it has the same smell as it was cooking, or as it is in the raw state, or if it's stored for a very long time, does it still smell fresh, or has it been spoiled? And one of the most important thing is about quality control, to make sure that the product is always consistent. And when the consumer or customer buys it, they know that it will be the same today as next week as next year. And that's all part of making sure the storage and packaging is the most optimal for each of these products. So how will this process be different in space? And gravity will be the missing ingredient in this process. And how will that change all the different steps I've just mentioned to you? Well, the first one is the cellular response. There are some advantages and disadvantages. One advantage I read is that there's a time dilution. That means the cells actually age slower in space. However, the disadvantages, due to the lack of research, it's unknown at the moment how the cells will grow or behave. And for the fish cells and the process of making the fish cells here in a bioreactor, we have to figure out how to handle liquids. So the mixing and transferring of liquids, they will always need this nutritional broth. And how do we keep them either mixed well within the broth, or how do we keep the cells within the broth in a container somehow? And then handling the cells. So they are in a suspension culture already, so maybe with space they can just be floating around. But we do need to separate the cells from this media or nutrition broth, because we want to only have the cell biomass as the ingredient and not the medium as well. And sterility. So how do we keep the sterility of all the liquids that we have, or even the handling? The operator will be floating, or can they be fixed? And the things that they need to use, how will they be fixed so that they're not floating around touching each other and ruin the sterility? Because this process still needs to have a sterile environment, or else you risk a lot of contamination. And then analytical tools. A lot of the analytical tools available now are meant for use on Earth. And it might be um, the lack of thinking about it, but gravity also plays a role in how these analytical tools actually can function. And most of them require a lot of liquids and handling, and flow through of things. So gravity plays a big part in the function of these tools. So just to give you an example of how the cells grow in suspension with gravity, I have two videos here to demonstrate the suspension of liquid, and how So you see here, it's a shaking incubator. The medium is being mixed around to provide dissolved oxygen and CO2 to blend or diffuse into the medium here. And then if we do a stir tank bioreactor, as you can see, the liquid, of course, stays at the bottom of the vessel. And then there's these impellers stirring and mixing the cells around and also providing a dissolved oxygen capacity. So gravity is essential for these types of culture methods. And in order for this process uh, to be successful in space, we have to think about a new bioreactor system that does not limit itself to function with gravity. Not only do we have to think about the limitations that a small vessel, as I showed you earlier in the video, to function without gravity, we also have to think if we need to meet the demands of, let's say, all the space inhabitants, we need to go bigger in volume to do mass production. So then these bioreactors that are going to do mass production also needs to function without the need for gravity. And that might be the biggest challenge in bringing seafood into space. And with that, I'd like to thank you for your attention. And I hope you learned a little bit more about cellular agriculture and Bluu Seafood as well. And if you have any further questions, feel free to contact me at my email address here. And I would like to thank or acknowledge the uh, team members or the company members at Bluu Seafood. Here we are at Hamburg, our new facility, and hopefully anywhere in the world to provide you with the next cultivated seafood product. Thank you.